Hello class, and welcome to the next section of chapter 12, which, as a reminder, is all about enzyme kinetics, inhibition, and control. And in this video, we're going to focus more on enzyme inhibition, which is inhibiting the rate of a reaction that is catalyzed by an enzyme. And so as we talk about enzyme inhibition in this video, first we're going to look at Lineweaver-Burke plots, another way of representing a reaction kinetic, as well as three types of inhibition: competitive, uncompetitive, and mixed. And then in the next video, the last video, we will talk more about the control of enzyme activity. But let's get started here by looking more at about how we represent kinetic data. So as a reminder from our first video, we got a first look at this Michaelis-Menten plot, which is this hyperbolic plot that you see here that's representing how the reaction velocity changes as the concentration of substrate changes. And so we talked a lot about how this reaction can reach this theoretical Vmax that depends on the and the rate of the reaction depends on the concentration of substrate that we see here. And we also talked about how 1/2 Vmax is a really important rate that we look at to help us determine the Km of the reaction. And as a reminder, the Km is what tells us more about the binding strength between the enzyme and the substrate. So the lower the Km, the more strong binding we see between the enzyme and the substrate. So similar but different, we can flip the Michaelis-Menten plot around to arrive at a Lineweaver-Burke plot, and that's going to be a much more helpful way for us to look at enzyme inhibition, because with the Michaelis-Menten plot, we were approaching this Vmax asymptotically, but we never actually arrived there. And so it can be, if we want to actually use the Vmax when we're looking at the rate of the reaction, it can be more helpful to flip this plot around like we do with the Lineweaver-Burke plot, which is a linear plot. And it's called a double reciprocal plot because we took the Michaelis-Menten plot, which plotted V0 versus concentration of substrate, and we made a double reciprocal, right? So now we have 1 over V0 on the y-axis and 1 over concentration of substrate on the x-axis. And this helps to make that reaction, or that plot, linear. And so this ultimately can also help us estimate our enzyme substrate binding strength, because Km is involved in several different aspects of this plot. And so let's look more at the different parts of this plot. So like I mentioned, this is linear, right? And so it's going to follow that linear equation for a line, which is y equals mx plus b, which is shown on the top here. And so our y, in this case, is our 1 over V0. That's what's represented on our y-axis. Our x-axis, like I mentioned, is represented by 1 over concentration of substrate. And then we have a couple of other parameters that are really helpful here. So first we have our y-intercept, our b, which is actually going to tell us 1 over the Vmax. So if we can find the y-intercept of a Lineweaver-Burke plot, we can calculate the Vmax very easily, which is pretty different from a Michaelis-Menten plot. And then the slope, the m, right, is given by Km divided by Vmax, right? And so if we're able to measure the y-intercept and the slope of a line that's part of a Lineweaver-Burke plot, we can really easily calculate the Vmax and the Km. Something important to note here, we also have the x-intercept available to us, right? And the x-intercept can be helpful for calculating the Km because the x-intercept is -1 over Km. But something important to note here is that this is actually more of a theoretical value, right? Because on this area of the plot, we're at a negative concentration of substrate, which is sort of meaningless in real life, right? So this isn't actually a measurable component. And so a lot of times when we see a Lineweaver-Burke plot, we actually won't see to the left of the y-axis, right? And so then we can't really use the x-intercept if it's not part of our plot. But that being said, if you are given a plot that has an x-intercept on it, you are very welcome to use it to help you calculate the Km for that reaction. So let's start to look more into what this means, or how these values can help us. And we'll do that by looking at 3 different types of enzyme inhibition. And so we'll talk first about competitive, and then uncompetitive, and then finally mixed inhibition. So first we'll start out, like I said, with competitive inhibition. And so competitive inhibition is when we have an inhibitor that's going to bind at the active site of an enzyme. And this is going to interfere with the binding of the enzyme to its substrate, right? So we have essentially two competing reactions, right? We have enzyme binding to substrate, forming the enzyme-substrate complex, but at the same time, we have enzyme binding to inhibitor, forming this enzyme-inhibitor complex. And so if we have this enzyme inhibitor complex, the substrate cannot bind to the active site of that enzyme, right? And so this reaction cannot proceed to form the product. And so as this happens, we have this problem, right? So we have the enzyme is bound to the inhibitor, it cannot bind to the substrate, we cannot form a product. And so ultimately what this is going to do is two different things with our reaction. So the first thing it's going to do is increase the apparent Km, right? So we talked more before about how Km tells us the strength of the enzyme binding to its substrate. And so when a Km is higher, that means we have weaker binding of the enzyme to the substrate. And so that's essentially what's happening here, because as the inhibitor binds to the enzyme, it makes less enzyme available to bind to substrate. And so ultimately overall, we'll have less enzyme-substrate complex forming. And so that's going to make our Km seem larger, because we're going to need more substrate to be able to get to that 50% binding of enzyme and substrate, right? And so, but at the same time, though, this is not going to affect the Vmax, because if you do eventually add enough substrate, this reaction can proceed to form product at the maximum rate. You just need to add more substrate before you can get there. And so let's break that down into a little bit more of what that means. So first we'll look at an example of competitive inhibition. And so we have this enzyme called proline racemase, and what it does is it interconverts L and D proline. So we see L proline here on the left, we see D proline here on the right. And as this reaction proceeds using this enzyme proline racemase, it's going to proceed through this planar transition state. And what that means is that we have this, this particular carbon here, it's going to no longer be tetrahedral like we see here. It's actually going to be in this planar form, right? It's only forming 3 bonds. And so there are competitive inhibitors for this enzyme that resemble this planar transition state that also have this planar carbon over here. And they also have similar charge distribution and a similar shape overall to this transition state. And so these types of inhibitors, which we see down at the bottom, are going to compete with the substrate to actually bind to the enzyme active site, right? And so that's going to prevent substrate from binding to the enzyme and ultimately prevent this formation of the product. So what does that mean when we look at our plots, right? So on the left we have our Michaelis-Menten plot. On the right we have our Landweber-Burke plot. And let's see how they look different when a competitive inhibitor is added into this reaction, into this enzyme and substrate mix. And so if we look first at our Michaelis-Menten plot, the top line in black is showing the reaction as it proceeds when there's no inhibitor. And then the green lines are showing how the kinetic would look as we add increasing levels of inhibitor. So here we have some inhibitor added. This next line down, we have 5 times that initial amount of inhibitor added, and then finally 10 times. And so as you can see on this plot, the Km, right, we talked about how the Km is going to apparently increase, right, during competitive inhibition, because this inhibitor is making it harder for a substrate to bind, right? You need more and more substrate to get to that 50% of relative rate. And that's pretty easy to see on this Michaelis-Menten plot, right? So we have 50% of V naught. Pretend that's a straight line. And if we drop that down with our no inhibitor, we have this Km. And then as we add inhibitor, we have to bring that farther and farther out to get to that 50%. And so the more inhibitor we add, the higher our Km is going to get, the more substrate we need to make that reaction get to that 1/2 Vmax. At the same time, it's harder to see with this graph, but our Vmax is remaining the same. And so if you were to keep extending this line, this graph even further out to the right, eventually all of these lines would meet each other. All of these curves would approach that same asymptotic Vmax at the top at that 100% rate. But so again, that's a little bit, it's maybe easier to see the Km, but it's kind of hard to see what's happening with the Vmax in our Michaelis-Menten plot. And that's where our Lineweaver-Burke plots can be really helpful. Right, so let's break that down. So first, let's start with our y-intercept, which, if you recall, is going to be defined as 1 over Vmax. And we were just talking about how for this type of competitive inhibition, our Vmax stays the same. And so in that case, 1 over Vmax would also stay the same. And so you can see in both lines, one with no inhibitor, one with a competitive inhibitor, the y-intercept is the same, right? But we do see a difference here when we look at the slope, right? So when we're looking at the slope, that's going to be defined as Km over Vmax. And this also makes sense, right? We talked about how our Km is increasing, our Vmax is staying the same. And so if our slope is defined as Km divided by Vmax, if our Km, the numerator here, is increasing and our denominator is staying the same, overall that slope is going to increase, right? And that's exactly what we see here on the Lineweaver-Burke plot. And then since here we have our x-intercept drawn out, we should also consider that. So our x-intercept is minus 1 over Km. And so, like we mentioned, our Km is going up, and so we can see that as our shift to the right with our x-intercept when we add that inhibitor. So let's take that into a more practical example. So we have our orange line that's drawn over here. And this orange line represents the rate of an enzyme-catalyzed reaction that's in the presence of a competitive inhibitor. So I'd like you to take a moment and consider which of these other lines is going to represent the activity of the same enzyme that's in the absence of the inhibitor. So please pause the video and take a moment and think about it. Okay, so let's consider all these different lines. So as we recall, for our competitive inhibitor, our Km is increasing, but our Vmax is staying the same. So if we want to consider these different lines, right, we want to identify the other curve on this graph that has, and so this is in the presence of a competitive inhibitor. So in the absence of that inhibitor, right, we would expect to see a lower Km, but we would still expect to see the same Vmax. So we can identify two different lines on this graph, right, that have a lower Km. That's the blue one and the yellow one, right? And so now we have to think about which one of those is going to have the same Vmax as the orange line. And so that's going to be the blue line, right? The blue line is going to have a lower Km, but it's going to be approaching that same Vmax as we saw with the orange line. On the other hand, our yellow line is approaching a much higher Vmax, and so that can't be the right one. And our gray line has a higher Km, so that's going to be something that has even higher level of that competitive inhibitor as compared to that orange line. All right, so let's move on to our next type of enzyme inhibition, which is uncompetitive inhibition. So during uncompetitive inhibition, we have an inhibitor that's going to bind to the enzyme substrate complex, which we see here. So just as a reminder, for competitive inhibition, our inhibitor bound to the enzyme itself. Our uncompetitive inhibitor is binding to the enzyme substrate complex. And so this means that enzyme and substrate can bind to each other, but then the inhibitor also binds to the enzyme substrate complex, forming this three-part complex, the enzyme substrate inhibitor complex, which ultimately prevents the reaction from proceeding. And so this type of inhibition is going to be interfering with the conversion of substrate to product. At the same time, it's also actually going to increase the binding of enzyme and substrate to each other, which might be a little bit counterintuitive at first. So let's talk about how that happens. So you can sort of think of this formation of the so recall how we thought about before as our enzyme substrate complex is essentially being in equilibrium, right? Where we have our enzyme and substrate separate, forming our enzyme substrate complex. We also have now another equilibrium here where our enzyme substrate complex can bind with our inhibitor, forming the enzyme substrate inhibitor complex. And so you can imagine that as more of this enzyme substrate inhibitor complex formation is happening, we're also increasing the rate of this reaction of enzyme and substrate coming together to form that enzyme substrate complex. So ultimately, overall, we're giving this apparent decrease in the Km, right? Because we see this apparently stronger binding of enzyme and substrate because we have more of this enzyme substrate complex that's being made. It's just a lot of it is being then shunted over to make this enzyme substrate inhibitor complex, which is going to prevent this part of the reaction from happening, right? And so again, that's going to result in a decrease in Km because we're going to need less substrate concentration to form that same amount of enzyme substrate complex. And then something else that's also going to happen is we are going to have a decrease in the Vmax. Because we have less ability of formation of product, ultimately we're not going to ever going to be able to reach that same Vmax that we would have without the inhibitor present. So let's look at what that means with our plots. And so as a reminder, we have our decrease in our Km, and we also have a decrease in our Vmax during this uncompetitive inhibition process. And so once again, let's start out with our Michaelis-Menten plot. So the reaction kinetic with no inhibitor present is shown with this black line here, and the Km is shown if you go across here with our relative rate, dropping the Km down to here. And then as we add increasing levels of inhibitor, we see this dramatic decrease, this lowering of these lines, right? And so this graph, the Michaelis-Menten plot, makes it very apparent that our Vmax is decreasing, right? However, it's a little bit harder to see what's going on with the Km here because as the Vmax is changing, 50% Vmax is also moving, which makes it sort of harder to plot on this graph where the Km would fall. But we can sort of try our best here. So our original Km is here. For our first line right below that, we have maybe our 50% Vmax is maybe around here, so our Km has moved a little bit to the left. Then for our next one down, it's maybe over here, so we moved a little bit more to the left again. And so you can see this small but clear decrease, right, in the Km on the Michaelis-Menten plot. But if we turn to look at our Lineweaver-Burke plot, it is much clearer to see what's going on. And so once again, let's start with our y-intercept, right? So our y-intercept is going to be defined as 1 over Vmax. And so, like we said, our Vmax is decreasing. And so when we add our uncompetitive inhibitor, if our Vmax is going down, our 1 over Vmax is going to go up. And that's exactly what we see here. Our y-intercept is increasing. At the same time, if we look at our slope, we have our slope is defined as the Km over the Vmax. And so this makes it very clear that, like we mentioned, our Km is going down and our Vmax is also going down. But as you can see very clearly on the Lineweaver-Burke plot, our slope is actually staying the same. And this is because both our Km and our Vmax are decreasing, and they're decreasing in a way that's proportional to each other. So overall, the slope is going to stay the same. And then finally, we have our x-intercept, right? And so our x-intercept is negative 1 over Km. And so we talked about how our Km is decreasing. And so we're going to show this as our shift to the left. It's our negative 1 over Km. All right, so let's move on to our third type of enzyme inhibition, which is mixed inhibition. So mixed inhibition is kind of just what it sounds like, right? It's a mixture of both of those other types of inhibition that we just discussed. So with our competitive inhibition, we saw this inhibitor that bound to the enzyme, forming this enzyme inhibitor complex and prevented substrate from binding. With our uncompetitive inhibition, we had this inhibitor that bound to the enzyme substrate complex, so substrate was able to bind to enzyme, but then once the inhibitor bound, product was not able to be formed. For our mixed inhibition, we see both. We see a combination of this, where our inhibitor is binding both to the active site of the substrate, the active site of the enzyme, as well as to the enzyme substrate complex. And ultimately, this is going to be also interfering with the conversion of substrate to product. But it's going to have mixed effects on enzyme substrate binding because it's basically a mixture of those two types of inhibition, where one of those types of inhibition did affect enzyme substrate binding, but the other type did not. And so ultimately, let's see what this is going to do with our Vmax and our Km. Right? So for our competitive as a reminder, we had our Kn was going up, but our Vmax stayed the same. For our uncompetitive, we had our Km going down and our Vmax also going down. And so let's consider how that's going to affect what's going to happen in our mixed inhibition, which is a combination of both of those things. So first, let's look at the Vmax, right? So if we have a mixture of competitive and uncompetitive, for competitive, our Vmax stayed the same, but our uncompetitive, our Vmax went down. So if you were to blend those two things together, ultimately your Vmax is still going to be going down. On the other hand, if we consider the Km, for our competitive inhibition, our Km went up, but for our uncompetitive inhibition, the Km went down. And so for mixed inhibition, there are, unsurprisingly, mixed effects on the Km. It might go up, it might go down, or it might stay the same. And so that brings us to a specific type of mixed inhibition, which is called pure non-competitive inhibition. And as a reminder, non-competitive inhibition should not be confused with uncompetitive inhibition. These are two separate things. For non-competitive inhibition, that type of mixed inhibition, it's an exact equal mix of competitive and uncompetitive inhibition, in which the inhibitor is going to equally bind to the enzyme and to the enzyme substrate complex. And this type of non-competitive, pure non-competitive inhibition is going to have no effect on the Km, because if it's an exact equal mix of Km going up and Km going down, then Km will stay the same. And again, this purely non-competitive inhibition will still have this decrease in the Vmax. And so let's take a look at what that is going to be like when we look at our plots. And so as a reminder, our Vmax is going down, but our Km is, we're not sure, could go either way. So once again, we have our Michaelis-Menten plot. We have our uninhibited kinetic with this black line that is approaching our Vmax at 100%. And then let's take a look at what's happening with our Km. So our uninhibited reaction, our Km, is going to fall right around here at our 50% reaction rate. But for our other curves, again, it's really hard to see what's happening here with the Km, similarly to how difficult it was to see that with our uncompetitive inhibition. And so for our next line down with the inhibitor, maybe it falls around here, maybe this one is falling around here. But it's hard to see what's going on, right, with this Michaelis-Menten plot. But it is still pretty easy to see that our Vmax is decreasing as we add more and more inhibitor. So let's take a look now at our Lineweaver-Burke plot and the differences that we see there. And so once again, our y-intercept in our Lineweaver-Burke plot is going to be 1 over Vmax. And so we were just discussing how Vmax is going to decrease when we add our mixed inhibitor. And so that's going to make the 1 over Vmax go up, just like we see here when our inhibitor is added. We also have our slope. So our slope is Km over Vmax. And so what's going to go on with the slope? So we just mentioned how our Vmax is going to go down. The Vmax is the denominator of this fraction, right? So as the Vmax decreases, the slope is going to increase. We also talked about how we're not sure what's going to happen with the Km, right? So Km might go up, might go down, and it's the numerator of this fraction. So if Km is going up and Vmax is going down, that will definitely always increase the slope of this line, right? We have Vmax going down, but if Km remains the same, it's still going to increase the slope of the line. And then if our Km goes down while our Vmax goes down, the slope of the line is actually still going to increase because the Km will not decrease as much as the Vmax is decreasing. You have a larger decrease in Vmax compared to the Km. So ultimately, our mixed inhibitors are going to have a slope that's larger compared to when there's no inhibitor present, like we see on this plot. And then finally, what we see when we look at our x-intercept, that gives us a pretty much clearer picture of what's happening with the Km, right? And so our Km, as you can see very clearly now from our x-intercept, does not change whether or not the inhibitor is present. And so that's what tells us for sure that this is a pure non-competitive inhibitor because our Km is staying the same. So hopefully that was clear for everyone, and so now you should be ready to go ahead and start answering the practice questions about enzyme inhibition.